welcome to Convergent Dialogues. This is Xavier Bonilla. In this episode, I'm very happy and very honored to be talking with Thomas Cech. Thomas is Distinguished Professor of Chemistry and Biochemistry at the University of Colorado-Boulder. He has been an investigator at the Howard Hughes Medical Institute since 1988, was president of that institution from 2000 to 2009. He has a PhD from the University of California-Berkeley and completed his postdoc at MIT. His main interests are in RNA and telomeres, and he receives the Nobel Prize in Chemistry with Sidney Altman, 1989, and also has the National Medal of Science in 1995. He is the author of the latest book, The Catalyst, RNA and the Quest to Unlock Life's Deepest Secrets. And that's what we talk about in the discussion. We start by talking about why RNA does not get discussed as much as DNA. We talk about some of the basics of DNA, kind of outline how it works. We talk about RNA and how it's a catalyst for many things. We talk about the splicing capabilities of RNA. We talk about transcription, translation, splicing. Talk about RNA as internal and external catalyst, origins of life. We, of course, talk about telomeres and extended life, different types of RNA, mRNA vaccines, CRISPR, and many other topics. Again, this is a big honor. Tom is obviously quite brilliant in his field. He's done so much work for understanding basics of chemistry and biology, understanding RNA, and a lot of his research and others has really helped us to have many wonderful advancements in medicine and within science. And so his work is just tremendous, and it was such a privilege to talk with him, and he was very, very kind and obviously very, very, very brilliant. As always, you can find this conversation and all other conversations at convergingdialogues.substack.com. I'm also on YouTube, so get over there, subscribe, share, follow, and contribute. And all of that is much appreciated. And now I bring you Thomas Cech. I am here with Thomas Cech. Tom, thanks so much for coming on the podcast. It's a big honor, big privilege. So really, really grateful for you doing this.
Happy to be here, Xavier.
Yeah, absolutely. So you've written a really wonderful book called The Catalyst, RNA and the Quest to Unlock Life's Deepest Secrets. And of course, you've had some wonderful work over your career. So we'll talk all about RNA. Before we do, just tell listeners kind of who you are, snapshot of you professionally, academically, and what you're currently doing.
I'm a professor at the University of Colorado in Boulder. I run a research group and do some teaching. And in my spare time, I like to hike in the mountains that are behind my house and go skiing, although that season is over for the moment.
You live, Colorado's beautiful. You're out there in the Rocky Mountains. Over here in the East Coast, we have the Appalachian Mountains, which are nice. And I live in the Appalachian Mountains, but I'll be honest, not as nice as the Rockies. The Rockies are quite gorgeous, so you're very lucky.
Well, any mountains are better than no mountains, as far as I'm concerned.
I firmly agree with you on that. So RNA. So, obviously we'll talk about why people know a little bit more about RNA, specifically mRNA, more recently in the past couple of years, of course, with vaccines and everything. And so a lot of people might have a little bit more kind of ears to RNA. But tell us, I guess, why do you think it is for a long time we had this emphasis on genes and DNA? DNA is all DNA and genes. But people kind of have neglected the role that the cell plays, the role that RNA plays. Why do you think that many people, at least in kind of, popular science or when people talk about things in big pieces, we haven't really talked about the mechanisms or the elements of RNA as also being important?
Well, DNA, of course, is critical for life. It stores all of the information that's required to build a human body and to keep it running. And because RNA is copied from DNA and has the same information content, but it's more of a temporary copy. It's not as stable as DNA. People have underestimated it. If it's just a messenger, if it's just a temporary copy of what's already in the DNA, then why do we have to really understand it? And that's changed a lot, both with finding of new aspects of messenger RNA, but also with new and various functions of ribonucleic acid RNA. So, to put it in another way, DNA has been the diva singing in the spotlight, and RNA has been thought of as sort of a backup singer, slaving away in the shadows.
Yeah. That's a good way to put it. So I want to talk about a little bit kind of the differences, but just kind of for listeners, just briefly, you kind of give us an overview. We have these A, T, G, C base pairs for DNA. DNA encodes proteins. Just kind of give us the snapshot, if you will, of how that works and where RNA comes into play as that backup singer.
So all this information that's stored in the form of DNA and RNA, the purpose of it is to build proteins. Proteins are the movers and shakers in all of our cells. They are the enzymes that digest our food in our stomach. They make our heart beat. They make our muscles move. And they allow information to be carried in the neurons in our brain. So how does this work? Well, the A, G, T, and C in the DNA are 4 bits of information, sort of like the zeros and ones on a computer program, but for DNA, we have 4 bits of information. Now, proteins are strings of amino acids like lysine and arginine and tryptophan, many of which we've all heard of. So how does this all work? So there are 3 bits of information in the DNA that encode a particular amino acid in a protein. For example, ATG means methionine, and TTT means phenylalanine. So that's the code. It sounds easy now, but it took a lot of work in the 1960s to figure that out. And the RNA copied from the DNA has those same 4 bits of information, but instead of A, G, T, and C, we have A, G, U, and C, where U is informationally identical to T. So it's really the same information.
You mentioned, you kind of give a little bit of history about Garnow and Watson, but you explain how RNA is single-stranded while DNA is double-stranded, and so they both speak the same language and just in a little bit differently. Is there any reason why we know that one gets the double and one gets the single if it's very much the same kind of material and speaking the same language?
That's right. Good point. Informationally, you wouldn't need to have 2 strands in DNA. 1 strand would be enough. But by having 2 strands, then when the DNA gets copied, the two strands get separated, much like when you pull down the zipper on your jacket and the 2 halves separate from each other. And now either half can be used to template or to specify the sequence on the other half. So the double helix allows for an easy way of copying, or we call it replicating, the code in the DNA. So one of the 2 strands of the DNA would have exactly the same information content as the RNA, the same orders of A's, G's, and C's going down the zipper, so to speak. And the other strand has the matching or the complementary sequence.
So when it comes to RNA, how is it kind of the thrust of the book, as kind of the title there is that RNA isn't just the messenger, although I think it can be that obviously, but it's the catalyst for many important life functions. I see it as a kind of like maybe spark plugs or an engine starter or some whatever analogy you want. It really can be a kind of catalyst here. How does that work? How do we know that it has those features? It's not just this kind of messenger that we commonly know it as.
So this first came to light in the 1980s, and my own research laboratory certainly played a role in this revealing of these additional functions of RNA, particularly the fact that it could speed up in a very specific way biochemical transformation in living cells. We found the first example of this in Tetrahymena, a pond animal found in freshwater ponds around the world. We didn't know how general it would be at the time, and we can talk about that discovery later. But that turned out not to be the most important place where RNA is a mover and shaker in cells. The most important place is really in what's called the ribosome. Mm-hmm. So these are the millions of protein synthesizing machines that are found in every living creature, from bacteria to humans. And they read out the code in the messenger RNA and spew out the matching string of amino acids in a protein molecule. And it turns out that these little machines that do this wondrous transformation, reading out the mRNA and making the protein, are themselves composed of RNA and protein. So it's a little complicated. It's using RNA to make protein, but the machine that does it is made out of RNA and protein. The RNA is called ribosomal RNA because it's not the messenger RNA, it's the RNA that's doing this job. And it turns out through work of Harry Noller and at UC Santa Cruz, and then later Tom Stites and Ada Yonath and Venki Ramakrishnan, we've learned that the RNA in the ribosome is really the machine. This is an RNA, an active RNA unit. So really the most important, one of the most important things in life, which is making proteins in a specific way, is itself catalyzed by RNA.
It's really fascinating how it's kind of you're talking about the ribosomes it's made up of that, but then it's also using that. It's just really interesting.
Yeah, and it took a while to figure that out because the proteins are playing a supporting role in those ribosomes, and so if you tried to get the whole process to work with just the RNA, it wouldn't work. So it's a collaboration between ribosomal RNA and proteins. So it took quite a bit of work to figure out that it was the RNA that was really the heart of the heart and soul of the beast, and the proteins were just supporting players.
And you mentioned early on, which was really interesting, and I'm curious for you to kind of expand on this, is this discovery of splicing, right, of for mRNA, and that it wasn't always a straightforward copy of DNA, and that it has many other features here for living organisms. Talk about this splicing of RNA and why that was such a critical find.
Yeah, this is really a thrilling story because it happened in the 1970s, and the earliest biological researchers who were interested in the fundamental molecules of life were looking at bacteria like E. coli. And they found that the messenger RNA was just a straightforward copy of the information in the DNA. And that made so much sense that the RNA would just be a straight copy that when they started looking at human genes, they fully expected to see the same thing. But instead, the human genes turned out to be much larger than the bacterial ones, even if the protein that was going to be the final end product was of similar size. And that was perplexing. And it turned out that most of our genes and most of the genes of all the plants and animals we see around us have the coding regions interrupted by stretches of non-coding DNA that are called introns. And you can think of this as a commercial message interrupting your favorite film. You really want to just fast forward through this stuff, right? And get back to the story. But there's no way that our cells can fast forward through these interruptions. And by the way, we don't know why they're there. We don't know really how they got there. Scientists are still arguing about this. But we do know what the current situation is. And so these interruptions are there in the chromosome, in the DNA. And now, when the DNA is copied into RNA in humans, these interruptions, these introns, are copied along with the coding sequence. So you get a gigantic initial RNA copy that's just a straightforward copy of what's in the DNA. And then this has to be cut and spliced, just like a sailor might take a frayed part of a rope and cut it above the fray and below the fray and splice the two ends together. I don't know if we probably these days we just go buy a new rope, but anyway, that's what the ancient mariners presumably did. And so this is what has to happen in all of our cells, right? We have to get rid of these introns so that we can have a useful messenger RNA. And this is the process called RNA splicing. And you might think it's just sort of an annoyance and something the cell has to deal with. But it turns out that human biology has made incredible use of this splicing process. Because if it always went that you splice from point A to point B and then to point C, there wouldn't be any added advantage in having these interruptions. But for most human genes, the RNA can be spliced in multiple different ways. Point A can be spliced to point B, or it can be spliced sometimes to point C. And this means that a single gene can give rise to multiple messenger RNAs, each of which encodes a related but not identical protein. So it gives us a lot more bang for their buck in the human DNA than you would have in a bacterial system that doesn't have these introns.
Is that what makes partly finding, all of the genetic properties for even something like height or eye color somewhat challenging? People will say, well, and then when you start scaling up to behaviors and other things, is because genes don't just stay kind of static in one place. There's all this types of business going on with the RNA, with splicing, with all these other things, such as with transmission and other elements. Is that what makes some of this kind of complicated to find? Well, here's the 600 genes for whatever. It doesn't quite work that way, right? It's not a one-to-one.
It really does complicate it quite a bit, because you would think that if you would have the same gene in your heart and your brain and your skin, that it would be doing exactly the same thing. But because of this so-called alternative mRNA splicing, it might be giving rise to one type of a protein in your skin and a related but somewhat different protein in your brain. So that certainly does contribute to the complexity of human genetics.
Now, I know that I'm a little fuzzy on this, but many people will understand RNA to have these elements of transcription, translation, transmission, and replication. How do you see, and now you're adding splitting in or splicing in here, excuse me. How do we keep all this straight? What's the order and sequence here?
Right, and I think your transmission must be from an auto show. We don't have transmissions in we don't use that word, but the other ones were all right. So let's put them in order. Okay, so the first thing is just transcription, and think about an ancient monk copying one document on papyrus into another copy, right before we had photocopying machines. Okay, so transcription is DNA into RNA. So that's transcription. Then translation means, if you think about the word in everyday language, it means that you're doing the going from one language to another, right? So now, in terms of RNA, you're going from the language of RNA, which is A's, G's, C's, and U's, to the language of proteins, which is lysine, methionine, tryptophan, et cetera, the amino acids. And so that's what happens on the ribosome, is that we do this translation from the language of RNA to the language of protein. And then the splicing has to occur in the middle there. It occurs after the transcription, but before the translation.
Yeah, it makes a lot of sense. Now, you had talked about, what was the bacteria that you were talking about earlier? That was the biocatalyst?
It's E. coli, but it's not the same one that we get sick from in fast food restaurants. It's a different strain of E. coli that is not pathogen. That's not a pathogen.
Well, in this part of the book, you talk about, let me see if I get this right, this RNase P, which is a ribosome important for understanding that RNA is an internal and external catalyst, which I thought was interesting how it's working on the inside and outside. Is this what you were mentioning earlier about the ribosome having its own thing and also needing to use RNA as well? Or is this something different? Tell me about this internal, external catalyst thing.
Right. Okay. So the first example of RNA catalysis, where pure RNA was able to promote a specific chemical transformation, came from our work with tetrahymena, and then Sidney Altman and his laboratory at Yale University found that another enzyme, not very much related to what we were working on at all, called RNase P. P stands for processing. Also was a so-called ribozyme or ribonucleic acid catalyst. So I think a simple way to think about this is that our system in tetrahymena, the RNA was undergoing self-splicing. It was cutting and pasting parts of itself together, and whereas the RNase P was working on external molecules. And an analogy for this, you talked about the internal and external. So our ribozyme in tetrahymena is sort of like tying your own shoes, right? When you tie your shoes, you tie two shoes and you're done. The RNase P that Altman found was like tying someone else's shoes. So you can do this continuously, right? You could tie, if you had the patience, you could tie 100 people's shoes or 1,000 people's shoes. And so this really extended the concept of a ribozyme from being just an internal rearrangement of an RNA molecule to something that could work externally on other RNA molecules.
Yeah, it's very, very interesting. In the book, you talk about these various people that every other chapter, it feels like, is someone discovered this, someone's doing this, someone found this. It's just such a interesting time in the 60s and 70s and 80s and early 90s where people are continuously discovering basic things about our biology, which is really fascinating.
Exactly. Things that were hidden that were there all the time, but we just didn't know about them.
So you had this really interesting chapter in the book. It kind of surprised me when I was reading it. You kind of cover a lot of this ground, and then you ask, which is the ultimate chicken or egg question. You say, so the origins of life, where does that come from? And I was like, I was surprised to hear this question. And you framed it really nicely. You said the better framing of it might be, how did the first entity capable of replication and evolution arise? Which is really, really fascinating. What did come first? Do we get DNA first, or do we get proteins first? What do we think, if we can possibly know? Was it always this way? Is like this fundamental to having life in this way? Life looked different. It looks different other places. How do we know how it evolved? How do we understand this process?
Well, it is such a absolutely bedrock fundamental question. So there's been a lot of conversation about it. And we don't know for sure because we don't have, there was no video surveillance of the origins of life. This is 4 billion years ago.
Right.
And, fossil evidence is so valuable for looking at later stages of living organisms, but molecules don't leave fossil evidence. They're too small to be fossilized. So the way that scientists have approached this is saying, well, let's try to recapitulate this in the laboratory. Let's try to see if we can make life from fundamental chemicals and that could have been around 4 billion years ago in the atmosphere. And if we can get it to work in the lab, then at least it's plausible that it could have occurred that way 4 billion years ago, although frankly, that's a historical question rather than a scientific question. So we'll never absolutely know for sure. So then getting back to your question, what came first, the DNA, which would be, you need some information to be passed down from generation to generation, or you wouldn't call it life, right? If it just existed once and died out, that's not much of a life form. So there has to be passing down of information to the next generation. But when we think of informational molecules, if you think of DNA, it doesn't do anything. It just sits there. Well, I mean, it does something, it stores information, but it's not active, right? It's totally passive. And it takes protein enzymes to pull apart the two halves of the zipper and make two new DNA molecules out of the one parental molecule so that you can go from a parental cell to daughter cells. So, okay, now things are really complicated, right? If at the origin of life, we had to have, by random chemical processes, DNA and protein enzymes to copy the DNA, all magically appearing at the same place at the same time, sort of good luck with that, right? That seems like a tall order. But now we know that ribonucleic acid can be both an informational molecule, like messenger RNA, just as good as DNA, at least in the short run, and it can be a biocatalyst, and it has the ability to string together individual building blocks of RNA to make bigger RNAs. Then maybe at the origin of life, there was just RNA doing both information and function, and this is the so-called RNA world hypothesis.
That's very, very interesting. I guess it's such a mystery, right? Because we don't have CCTV from 4 billion years ago, right? We can't look at what happened and how it all started. But it would be very helpful, wouldn't it, to get some of these answers sorted out. One of the things that's interesting in the latter half of the book, you talk about various things, obviously COVID-19 and the vaccines, which we'll get to in a minute. But you talk about telomeres. And extending life. And what's the story here with, you can tell us what they are and how RNA is used to work with chromosomes and how extending life at the cellular level could or could not, or there might be something there for extending the life of the organism as a whole.
Yes, this is a topic, this sort of fountain of youth topic has certainly caught the imagination of the general public. In fact, you can go on the internet and you can find products that are at least claimed to lengthen your chromosome ends or your telomeres, which supposedly, according to the bottle, would have a longevity enhancing effects. We'll get back to that in a minute. But what are these chromosome ends? Okay, so each chromosome is a very long molecule of DNA, but it's not a circle. It has two ends, and these ends cannot be copied when you go from the mother to the daughter cell by the same machines that copy the middle parts of our chromosomes. Instead, there's a specialized machine called telomerase, which builds out the ends of the very ends of the chromosomes. And this was discovered by Liz Blackburn, Carol Greider, and Jack Szostak, who got the Nobel Prize for their work on telomerase. So what does this have to do with longevity? Well, it turns out that this telomerase enzyme, which is active in our sex cells, in our germline cells, which is the technical term for the sex cells, and also in our stem cells, by keeping the ends of the chromosomes intact, it allows the cells to keep dividing continuously. However, in most of our adult cells, like in our skin and in our neurons in our brain and our heart cells that are beating away, this telomerase enzyme has been turned off before we're even born. And now the chromosome ends can't be maintained, they can't be copied. So the chromosomes actually shrink a little bit on their ends every time the cell divides. And this serves as sort of a yardstick for measuring how many cell divisions a particular cell is allowed. Because when these chromosome ends or telomeres get critically short, this signals the cell to stop dividing. Now, if you think about it, this is sort of a good thing, right? I mean, once you're an adult, you don't want all of the cells in your body to continue to divide. You want the division to be limited to cells like stem cells that need to rejuvenate some of our worn out tissues. So I think there's been a little bit of hype and a little bit of misunderstanding in some of the popular literature that people think if we could just keep our cells dividing, we could live forever. But there's a difference between cellular level immortality and organismal immortality. And I think if we just had all of our cells being immortal, we would be extremely large. And we would just be growing without bounds. And that would not be a good thing, because another definition of a human cell that has unlimited proliferative capacity is a tumor cell. This is a property of cancer. And so, in fact, telomerase is activated in human cancers. It's sort of a common denominator of most human cancers. So being you have to be careful what you wish for if you're wishing for immortality, because cancer is also an example of an immortal cell.
Yeah, that's a good
Sorry
point.
about the long explanation.
No, it's very good. It's very, very informative. You also mentioned therapeutic siRNA. So before you tell me about this, the breakthrough and understanding things about biomedicine here, some people may be wondering, we haven't maybe stated it explicitly, but there's different types of RNA. We got messenger RNA, which is mRNA. We have transfer RNA, which is tRNA. We have rRNA, a ribosomal RNA. And now you're introduced, I know there's other ones too, is HNRA, RNA, and then you're introducing siRNA. Can you tell us the significance of, or what the taxonomy is here a little bit, of all these different, like you get the small letter, the before the RNA in all caps. So what's the taxonomy here?
Yeah, well, thank you for that question, because I think I could have said this earlier. Let's just step back and think about there are really just two kinds of RNA, two big categories, right? So there's the message, which has the codons that encode the various amino acids in the proteins. And then there are a large number of so-called non-coding RNAs. And so these are RNAs that don't care about the genetic code at all, because they do wondrous and diverse other things in cells. So all of these other RNAs that you talked about, the RNase P RNA that we talked about, the tRNA, transfer RNA that carries the amino acids into the ribosome, the ribosomal RNA, and now the siRNAs. These are non-coding RNAs. And that's why the RNA field is so much more nuanced and is so rich compared to DNA, because DNA is just storing information. RNA has all of these non-coding roles as well as its coding role.
Mm, mm. Yeah,
So
that's very.
do you want to do siRNA then?
Yeah. Briefly, just tell us why that's why the therapeutic value of it, and why it could be, what the breakthrough was for biomedicine.
Yeah, it's stands for, and as you said, it's two small letters and then the three capital RNA. So the SI is small interfering RNA. These little guys are only 23 units of RNA long, 23 A's, G's, C's, and U's, and they match some other sequence of RNA so that they can pair with it. And when they do this, they cause that other RNA to either be cut up or at least not to be translated into protein. And so this is a natural process, and it was first found in these tiny roundworms called nematode worms. The scientific name is Caenorhabditis elegans. So they are very elegant little worms, and it's a natural process. So how could that possibly be of medical interest? Well, as often happens, because everything is connected towards evolution, what goes for the worm goes for the human, and we have this same process going on in all of our cells, so that when you add this siRNA to a human biology system, the machinery to make use of it's already in all of our cells. And they're just waiting for the instructions encoded by this little RNA about which messenger RNA they should attack using this siRNA as sort of like dropping a pin on an internet map, sort of where should we do our action, right? And so what the machinery is looking for is a messenger RNA that matches this siRNA. And when the two pair up, now you think about my zipper analogy. Now we're zipping up the zipper, right? We're matching the RNA, the siRNA to a messenger RNA that's pre-existing in a human cell, and this will then inactivate the human messenger RNA. So if it's a disease-causing messenger RNA, then this can be therapeutic. And there's a company in Cambridge, Massachusetts called Alnylam, which has now 5 therapeutic modalities against rare diseases based on this principle.
Wow, that's it's really a marvel, right? It's incredible how we can find this, and what the implications it can be for treatment, is just incredible.
And it happens over and over again. What people people discover things in pond animals like tetrahymena, in roundworms, and very often the biology has been conserved through vast stretches of evolution, and it applies to humans as well. We'll see that again when we talk about CRISPR.
Yeah. Okay, so there's nobody better to talk about I think the vaccines that we had for COVID-19 than yourself, because you can tell us. I've heard like kind of the elementary version of this, or the kind of the McDonald's version of this, so give us the full explanation. We had a virus, and we had a global pandemic, and within a very short time, we can talk about that in a minute, some of the suspicions that people had about that, but we had vaccines, and there was a newer technology, these mRNA vaccines. Could you just tell it? I mean, you can set up the virus for us if you want, or viruses generally, if that helps. But can you just tell us, soup to nuts, how this works in terms of the vaccine, specifically with the mRNA and the spike proteins? A lot of nonsense out there on what is and isn't happening. Can you just kind of start to finish tell us how it works, what's happening at this level?
Sure. So you asked a lot of questions
Yeah,
there.
sorry.
So if I forget to answer six of them, please
Yeah, I'll
repeat.
keep you on track.
But so what is a vaccine? So a vaccine is a way of giving a heads up to our immune system and saying, if you ever see anything that looks like this, it's a bad thing, and you need to make antibodies against it, and you need to kill any cells that are showing this property, right? So it's just sort of like advance warning
Mm,
system.
mm,
And so, what is it about a virus that you would want to show your immune system to give it this advance warning? Well, not something on the inside of the virus. That's not something that's visible. So it has to be something that's on the surface of the virus, and that thing is different for every virus on Earth. Okay,
mm,
so for SARS-CoV-2, it was these roughly 90 spikes protruding from the spherical virus that is the distinctive feature. It's
mm,
not found in human cells, so therefore, if you expose our immune system to this spike protein and say, be on the lookout for this guy, it's
mm,
portending something bad, then when the SARS-CoV-2 virus comes along with those same spike proteins, the immune system will be ready to attack it. And because our own bodies don't have naturally spike proteins, it'll see this as a foreign substance, as something, as an invader, rather
mm,
than part of our natural biology.
mm,
So then the next question is, okay, how do you train our immune system to be on the lookout for the spike protein? Well, there's multiple ways. And there were, in fact, about 20 vaccines that went into development around the world. The mRNA vaccines just turned out to be the most powerful of those. But
mm,
let's just talk about the different ways you could do this. You could inject the an inactivated, let's say a heat inactivated SARS-CoV-2 virus into people. That's actually the kind of vaccination that we do for most common diseases. It's a little frightening because it is the virus that you're shooting into people's
mhm,
arms, although it's been tested for safety and it's been inactivated. Now, even better is to just give people the spike protein, because then there's no chance that the spike protein can't do anything without the innards of the virus, right?
mm,
Without the RNA that's on the inside of the virus. So if you just show people's immune system the spike protein, it's a safe way of immunizing them. So you could inject them with the protein, but what the very clever folks at Moderna in Cambridge, Massachusetts, and at BioNTech in Mainz, Germany, figured out was that if you just put in the message, the messenger RNA that encodes the spike protein, then the human body cells will translate that over and over and over and over again. And you can
mm,
get hundreds of spike proteins from one messenger RNA before its stability is over and it disappears. So
mm,
it's sort of a nice amplification factor,
mm.
and that turned out to work, really well. Now, some of your listeners are going to say, "No, it didn't work that well. I got the vaccine and I still got sick." Okay,
Mhm,
but you didn't die, right?
mm.
And so it turned out that these respiratory viruses it wasn't the vaccine's fault. These respiratory viruses are tough ones to vaccinate effectively against. And so, yes, sorry, we didn't protect you from reinfection. Hopefully, we protected you from hospitalization. And the estimate is that about 15 to 20 million people would have died if they had not gotten the mRNA vaccine around the world.
Is that also because, as a virus will do, it's trying to live, it's trying to survive, and it kept mutating. So we had all of these variants that were, kind of the It's like when you update your phone, right? It's like, oh, this is a little bit better. It's kind of the same thing, but it's a little bit better, a little bit meaner, leaner, and faster. Obviously people will remember, the Omicron variant and all the other ones that were after that. Is that what made this a little bit difficult with the vaccines, where it was like the basic idea, it can ward it off, but the kind of meaner, cleaner version, so to speak, from the virus perspective, is a little bit tougher. You're not going to die, but you still might get sick. Is that a kind of very
you
pedestrian way of saying that?
Yeah, you said it well. When these RNA viruses copy themselves into their progeny viruses, they make mistakes, and they're pretty sloppy copiers. And if they're too sloppy, then they make inactive copies of themselves, and they do that, and those just fade away, right? They have no infectious ability, but sometimes they just by good luck for them and bad luck for us, they make a copy that now makes a spike protein that has a little bit different shape than the parental spike protein. And now if we have a vaccine trained against the old version, and now the virus has this new version, it's much less susceptible to the vaccines that were raised against the initial virus. So absolutely, the fact that the virus is constantly spinning off subtle variants allows it to keep trying to evade our immune system. But the nice thing about the mRNA vaccines is that they're so quick to make that you can then read out the sequence of the new variant, Omicron or whatever it might be. You can say, aha, it's got 3 amino acids in the protein are different from the old version. Let's just change the mRNA vaccine. We know the genetic code. We know which groups of 3 bits of information on the RNA encode each amino acid. So any student in my laboratory in Boulder could write down for you within minutes on a piece of paper how we would have to change the code of the RNA in order to now attack the new version of the virus. Of course, it takes some months to actually make that vaccine and to get it approved.
I guess the one follow-up I have here is when people hear this, they might say, and the skeptic might say, well, if we're having the mRNA and we're having and through the vaccine and to create these spike proteins, what's the long-term effect if we have this in our body? Could this potentially, maybe it protects us against a virus, or it fights against it, but could it harm us in some way we don't know? Or what's the long-term effects here of putting this stuff in our body, and we don't know what could be the outcome of that or how it mixes with other things? Is that a legitimate criticism or a legitimate worry, or is it pretty, with how we know about biology and RNA, not really something that we should be totally concerned with?
So one thing worth knowing is that the mRNA has what we call a lifetime. In other words, how long does it last in our body of about 1 day. And that's, in fact, why it has to be kept at such ultra cold temperatures while it's being stored and shipped, because it's not real stable. We actually wish it was a little more stable than that. But in terms of safety, it is, I think, reassuring that it does not stay in your body, nor does the spike protein continue to be around. It trains the immune system, and then it's gone. So maybe that's the good news for safety. It's also a bit of a bad news because it means you have to get a booster shot. You have to get reinoculated every year, just like you do against the flu. Now, a DNA vaccine would be very long-lived, right? We can even recover DNA from Neanderthals or from woolly mammoths and sequence it. So DNA is incredibly stable compared to RNA. Well, again, there's a plus and a minus to this. If you vaccinated with DNA, you might be able to have sort of a permanent vaccination. On the other hand, you've got something in your body that could potentially crawl into your chromosome and become part of your genetic makeup. And that might be harmless. It is a bit of a safety concern. So the RNA is considered to be really an ultimate in terms of safety. The other thing I think that, and I would encourage people to go to the CDC Center Disease Control website, where there's information about the safety of vaccines, like the flu vaccine that have been given to millions.